Good morning, good afternoon, or good evening. This is Biology 205, Anatomy and Physiology 1 at Illinois Central College, and my name is Lisa Whitehead. Today we are studying Chapter 16 in our text, the Autonomic Nervous System. Today's learning outcomes include the following. To compare the functions and the organization of the autonomic and the somatic nervous systems, we will review the pathway of visceral and motor responses as they travel from the spinal cord to the ventral root and then beyond, and that's from Chapter 13. We'll describe the dynamics of a sympathetic response of the autonomic nervous system. So this will include the sympathetic chain ganglia, collateral ganglia, and the adrenal medulla. We'll compare the classes of sympathetic receptors, including both alpha and beta. And we will list the neurotransmitters that are involved. And this includes preganglionic, postganglionic, and nitric oxide. Also, we'll describe the dynamics of a parasympathetic response of the autonomic nervous system. So we'll discuss differing body responses. So for example, what happens when the parasympathetic division is activated? We'll discuss where are there parasympathetic synapses. And we'll discuss the anatomy and the function of the vagus nerve, which is cranial nerve 10, and the importance of its output and stimulation. We'll also list the neurotransmitters that are involved in a parasympathetic response. Then we'll go on to describe the relationship between the two divisions of the autonomic nervous system and the significance of dual innervation. We'll discuss the significance of autonomic tone. We'll describe the difference in structure and the effect of long and short visceral reflexes in the autonomic system. And then we're going to start talking about memory. So we're going to differentiate between fact versus skill memory, scanning versus short-term versus long-term memory, and we'll define the role of the hippocampus in our memory. In today's lecture, we're going to discuss the autonomic nervous system, which is sometimes referred to as the visceral motor system because our autonomic nervous system is actually sending information to our visceral organs. So all of these visceral organs operate outside of our awareness, so they're operating without our conscious instruction. The autonomic nervous system is going to control all of our visceral organs, or visceral effectors, and they'll coordinate our cardiovascular function, so how fast our heart beats, for example, if we vasodilate, vasoconstrict in response to stress, or if we're relaxing. Similarly, our respiratory function, whether we have a deeper respiration or a more shallow respiration, what the rate of our respiration is. All of these things, of course, have an incredibly important function in our ability to respond to an emergency, or when we relax, we have the exact opposite type of response. Also, this will coordinate our digestive function like peristalsis, our urinary function, and our reproductive functions as well. Later on in the chapter, we're going to discuss higher order functions like consciousness, learning, and intelligence. And then after that, we're going to talk briefly about aging and the effect of aging on the nervous system. As always, today we're going to proceed through the content in the order in which is presented in the textbook. So that way, if you ever want to go back and review a certain area, it should be easy for you to find. So we're going to start off today talking about the divisions of the autonomic nervous system. We'll then discuss the sympathetic division, then sympathetic neurotransmitters and receptors. Then we'll move into the parasympathetic division and talk about parasympathetic receptors. Following that, we'll discuss the effects of the organization of our autonomic nervous system. We'll discuss dual innervation and what that means, the autonomic regulation and integration. Finally, higher order functions, which includes things like our learning, intelligence, and consciousness. And then lastly, we'll wrap everything up with a discussion on age-related changes in the nervous system. In previous lectures, we've already discussed the somatic nervous system and how it has control over things that are voluntary, specifically our skeletal muscles. So we can choose to pick up a cup of coffee or kick a soccer ball. These are things that we can voluntarily control. But in today's lecture, we're going to omit that and we're discussing the autonomic nervous system, which is controlling all of our viscera. So our viscera has effectors that is controlled involuntarily. So things like smooth muscle, whether that be smooth muscle in our uterus that we cannot stop contracting, although we may like it to, or, for example, smooth muscle that lines our blood vessels. These are things that we cannot control. So they are controlled by the autonomic nervous system. Other things that are controlled by the ANS include our glands, our cardiac muscle, as well as our adipocytes, or fat cells. The hypothalamus plays a role here in that it contains integrative centers, and the neurons here are comparable to those we already discussed previously when we were looking at the somatic nervous system. The upper motor neurons would have the same sort of role. And so the motor neurons of the central nervous system are going to synapse on the visceral motor neurons in autonomic ganglia. We're going to discuss this more coming up. In the autonomic nervous system, the visceral motor neurons can be broken down into either preganglionic neurons or postganglionic neurons. So the preganglionic neurons are exactly what the name infers. They are before the ganglion. So the cell body will arise in the brainstem or spinal cord, and then the fibers, which are just the axons of the preganglionic neurons, are going to extend from the brainstem and the spinal cord to the ganglion. So after it leaves the CNS, they synapse on the ganglionic neurons, also known as the postganglionic neurons. And these autonomic ganglia are going to have many different ganglionic neurons, and they'll innervate all of our visceral effectors. So the postganglionic fibers are going to be those that extend from the ganglion out to the visceral effectors. So we're going to discuss this more coming up, but just for now, understand that the preganglionic neurons are the equivalent of our upper motor neurons, and the postganglionic neurons are the equivalent of the lower motor neurons, if you want to draw that comparison. In your textbook, figure 16-1 is going to show you the organizational breakdown of the somatic and autonomic nervous systems. So let's compare these two, because although they're very similar, there are some differences. Let's start off with the somatic nervous system. This is on the left because we've already covered it in previous lectures. So you're already familiar with how the upper motor neurons are going to arise out of the primary motor cortex in the cerebrum. So you can see at the very top of the image, we have the cell bodies starting in the primary motor cortex, and then the long red extensions are there to indicate the axons of those upper motor neurons. So those upper motor neurons are going to descend from the primary motor cortex of the cerebrum and then synapse in one of two places. So on the left, you'll see that it synapses on the somatic motor nuclei of the brainstem, and then the example on the right has the axon extending all the way down into the spinal cord, and then it synapses on the somatic motor nuclei of the spinal cord. In both of these situations, once those upper motor neurons synapse on those locations, then the lower motor neurons are responsible for carrying the information out to the skeletal muscle, of which we have voluntary control to allow us to move. So let's compare this with the image on the right, which is the autonomic nervous system. So the similarity here is that we have, starting in the central nervous system, we have our first motor nuclei, so we'll call these our visceral motor nuclei. They originate in the hypothalamus, so you can see that that's where the cell bodies are. And then the purple extensions are the axons extending down to where they're going to synapse. So on the left, you see that that axon synapses in the autonomic nuclei in the brainstem, pardon me, and then on the right, it synapses on the autonomic nuclei in the spinal cord. Either way, now what we have next is we have a preganglionic neuron that inserts itself here. That preganglionic neuron is the neuron that takes information from the central nervous system and then extends to the ganglia. Then once it arrives at the ganglion, we have the ganglionic neurons, or the postganglionic neurons. Those ganglionic neurons are going to be responsible for taking information to all of our visceral effectors. So you see that little schematic on the left there showing you smooth muscle, glands, cardiac muscle, and adipocytes. It is from the ganglionic neurons that that information comes from. The autonomic nervous system has two divisions, the sympathetic division and the parasympathetic division. The parasympathetic division is for relaxation, and the sympathetic division is for action. So you've probably heard of the fight or flight response before, and this is what allows us to deal with an emergent situation. So, for example, if you are walking on your way to class and you are thinking about an exam coming up and some other deadline and what groceries you need to pick up on the way home, you have all of this on your mind, but at the moment that you see a car approaching at a rapid speed and you think that they might hit you, you're going to forget all of that upper level thinking immediately, and you instead will become very alert to the situation at hand to be able to respond immediately. So that sympathetic division is what kicks in to allow you to move quickly and to make a change to save your life. So it does this by increasing our alertness, also by increasing our metabolic rate, so we have lots of energy ready to go to be able to either run or fight or whatever it is we need to do to get ourselves out of harm's way. Then we also have the parasympathetic division. The parasympathetic division wants us to just rest and digest. So this is after a giant Thanksgiving dinner, how everybody feels tired. It's not just the tryptophan and the turkey. The parasympathetic division is also at play here. So the parasympathetic division will be responsible for helping us to conserve energy, and it helps to maintain our resting metabolic rate. Fun fact, by the way, my husband, whenever he works any type of night shift or has a all weekend long call schedule where he's out for 72 hours at a time, he'll actually not eat specifically, because if he does, then his parasympathetic nervous system kicks in and he wants to go to sleep. But if he keeps himself a little bit hungry, then he can continue to keep suffering through being awake all night. So just something to think about for some of you who might be working night shifts already or in the future in healthcare. The sympathetic and parasympathetic divisions of the autonomic nervous system have completely opposing effects. The sympathetic division is going to be that fight or flight response, so it energizes us to be able to respond to an emergency. The parasympathetic division has the opposite effect in that it relaxes things so we can digest food and heal. So while they have opposing effects, it's worth noting that the two can work together, but they can also work independently. So for example, that example I gave you before, if you're walking down the road and you're thinking about an exam and you see a car approaching and you think it might hit you, you forget about all of the things that you were thinking about that you had to do on your way home, and you think about what you have to do immediately. So the sympathetic division is kicking in to excite certain parts of your body, such as your skeletal muscles, so you can run away. And the parasympathetic division is going to kick in too, but instead of it activating things, it actually inhibits certain structures. So, for example, you just had a giant meal out at a restaurant, and you're walking and you see this car about to hit you. Your parasympathetic system will help you to stop digesting food, because you really don't need to focus on digesting that burger at that moment. What you need to do is run. So the sympathetic nervous system and the parasympathetic nervous system can kick in together, but they work as opposing features to the same end. Now, they can also work independently, and in that case, it's worth noting that in some cases, there is only one division that will innervate some structures. We'll talk about this coming up. So when these do work together, when we have the sympathetic and parasympathetic divisions working together, it's important to note that we don't have both the sympathetic division and the parasympathetic division both affecting the gut at the same time. What's going to happen is the sympathetic division will excite different parts of our bodies, and the parasympathetic division is going to affect other different parts. So they each control different stages of this whole process that allows us to take care of ourselves and remain alive. Let's continue with the example of you think you might get hit by a car as a pedestrian. What happens? The sympathetic nervous system is going to do all of these 7 things. It will heighten our mental alertness so you can figure out which way do I have to run. It will increase your metabolic rate so you have the energy ready to be able to have energy to run. You'll also have reduced digestive and urinary functions, which of course you don't need to be digesting food or needing to go to the washroom at the time when you might need to run away. It will activate some energy reserves so you have that energy quickly available to skeletal muscles, which are going to have a very quick demand as they need to run unexpectedly. Also, you'll have an increased respiratory rate, and your passages of your respiratory tract will dilate so that way you can take in even more air. So this allows you to have greater oxygenation and gas exchange, so that way again you're able to carry that oxygen to the different parts of your body, specifically skeletal muscle, right, that needs all that oxygen in order to continue reacting quickly. Your heart rate will increase, which makes sense. You feel like you're very tense and anxious. You may even feel it in your chest. Your blood pressure will rise because it's pumping that oxygenated blood out to all of those muscles so you can run, and your sweat glands become activated. So you can almost basically say all of these same things happen potentially with test anxiety. You can have the exact same experience. So the sympathetic nervous system is just responding to anything that stresses us considerably. The parasympathetic division of the autonomic nervous system is pretty much the exact opposite of the sympathetic division. This allows us to chill out and relax and heal and digest. So when all of those things are happening, we'll have a decreased metabolic rate because we don't need to have the same sort of energy to run a marathon as we do just to sit and watch TV or take a nap. We'll have a decreased heart rate and our blood pressure will come down. So we're calm and relaxed. And while we're calm and relaxed, then we'll have increased secretion of our salivary and digestive glands, so that will help us to digest our food and to extract nutrients from what we've ingested. We'll have increased motility and blood flow in our digestive tract. So this basically helps us with peristalsis, so that way food can be processed through from start to finish through our digestive tract. And we'll have stimulation of urination and defecation, which is why if you were running from a bear, you wouldn't think, I might need to go to the washroom right now. That would be absolutely only stimulated when you're relaxing. But if the sympathetic division is kicking in, then this is going to be exactly the opposite. This will be suppressed or inhibited. Let's discuss section 16-2, the sympathetic division. The sympathetic division of the autonomic nervous system is sometimes referred to as the thoracolumbar division, and we'll discuss this more in coming slides, but you'll see in the very next slide how this makes sense with a visual schematic. So what we have in the thoracolumbar division or sympathetic division are short preganglionic fibers that are in our thoracic and lumbar segments of the spinal cord, and these are going to be located between the segments of T1 and L2, so thoracic 1 and lumbar 2. The cell bodies are going to be located in the lateral horns, and the axons will enter in the anterior roots. So at this point then, we have the ganglionic neurons that are located in ganglia that are usually right next to the spinal cord. And then from that point on, we have the long postganglionic fibers that are going to stretch all the way to the target organs. So the short of it here is that in the sympathetic division, we have short preganglionic fibers, sometimes two, and then we have a long postganglionic fiber that stretches from the ganglion to the target site. Figure 16.2 in your text is one of the spotlight images. So it's a 2-page spread showing you on the left the autonomic nervous system broken down into the sympathetic division, and then the right page is the parasympathetic division. So we'll come back and discuss the parasympathetic division later. But for now, let's just look at why we call the sympathetic division the thoracolumbar division. So you can see here what we have are a whole number of different nerves kicking off of our spinal cord. So these are branching off from the levels of T1 through L2, and it looks almost like a ladder. And this is a perfect analogy for the sympathetic division. So as you can see, we have all of our preganglionic fibers illustrated in red, and they are going to synapse at ganglia. So then from that point on, once they've synapsed at the ganglia, there will be postganglionic fibers that are long, and they stretch all the way out to all these different organs to allow us to respond to an emergency. So just starting at the top for a quick example, you see the eye on the top right. In an emergency, if you thought a car was about to hit you, you would probably want to open your eyes wide so you can watch everything. Maybe blink a little bit less. Your pupils might dilate. All of these things would happen with your eye, so that way you could be maximally responsive to the situation at hand. Similarly, you have other different parts of your body that would need to respond quickly too. So you can see through the cardiac and pulmonary plexuses, we have sympathetic nerves extending to our heart and lungs that will allow us to breathe more deeply and oxygenate our blood, so that way we have lots of oxygen to be able to be transported throughout our body to our skeletal muscles, which will have an increased demand if they have to run immediately, for example. So you're going to notice a little bit lower down on the image that we have a few different sites that are kind of larger yellow blobs, and these are larger ganglion. So you can see at the top we have our celiac ganglion, then there's also the superior mesenteric ganglion, the inferior mesenteric ganglion, and we'll discuss all of these in more detail coming up. But for now, what I want you to take a note of is why it's called the thoracolumbar division. This starts at the level of T1, extends all the way down to L2. At the level of T1 through L2, you're going to see that the nerves arise off of the spinal cord. But running parallel on both sides of the vertebral column, you're going to see what's called the sympathetic chain ganglia. And these are a series of ganglia that are lined up in a row, and these look like a string of pearls, just because we have all these little fibers interconnecting one to the next. So what's going to happen here is we have our ganglia that have the ability to innervate any number of different body organs or group of organs, but we're going to have one preganglionic fiber that's going to come into that ganglion, and when it comes into that ganglion, it's going to synapse on all of the neuron bodies that are in that ganglion. So that allows that one arriving action potential that comes in through one specific preganglionic fiber to synapse at the ganglion, and then action potentials may be triggered to go to any number of different sites that are innervated by that specific ganglion. So you can see in the image here on the right, just for a very quick example, let's go down to the level of about T5 through T9. So we have here all nerves heading as preganglionic fibers extending out towards the right. They go through the greater splanchnic nerve as they converge, and then they're going to synapse at the celiac ganglion. So here at the celiac ganglion, the preganglionic fibers synapses, and then that information can be sent to a number of places. So you can see that the postganglionic fibers are here illustrated in black. So the postganglionic fibers can extend to things like the liver or the stomach or the pancreas or spleen. All of those are innervated by our celiac ganglion. So again, in the sympathetic division, we have our short preganglionic fibers that are in the thoracic and the lumbar segments, hence the thoracolumbar division. And then we have our ganglionic neurons that are located in ganglia next to the spinal cord. And then we have our long postganglionic fibers that are going to extend out towards their effector, usually a visceral organ here. So let's look at how this works just a little bit more detail. So those ganglionic neurons, these are the ones that have received information from the preganglionic fibers. The ganglionic neurons are going to then synapse in one of three different locations, and these are the only choices. It can either synapse in the sympathetic chain ganglia, which we just discussed, that's the string of pearls that you see on both adjacent sides of the spinal cord. They can synapse on collateral ganglia. So these collateral ganglia, for example, are things like the celiac ganglion that I just talked about in the previous slide. So they can extend through something like the greater splanchnic nerve and then synapse at the celiac ganglion, which is a collateral ganglia. And then they could also synapse specifically directly onto the adrenal medulla. The sympathetic chain ganglia, which is what looks like that string of pearls on both sides of the vertebral column. This is also known sometimes as the paravertebral ganglia. So these are going to be able to control all sorts of different visceral things like our body wall, our thoracic cavity, our head, neck, and limbs. Figure 16.3 in your text shows you the pathway of the sympathetic chain ganglia innervation. So in this image, this is just a schematic showing you what can happen in one of two different ways, but this could happen on either and both sides of the body. So what we see here is in red the preganglionic neurons and in black the ganglionic neurons. So let's look at the left first. So our preganglionic neuron exits the gray horn, and then it's going to synapse on the autonomic ganglion on the sympathetic chain. In this case it's on the left, but that's just an example. So it's going to synapse on that autonomic ganglion, and then that allows for the ganglionic neuron in black to carry that information to other distant sites, specifically directly to the effectors. So the example on the left is showing you the direct pathway from the preganglionic neuron to the ganglionic neuron, which then innervates visceral organs. If you look on the right, instead of going directly to the thoracic cavity, for example, and visceral organs by the sympathetic nerves, we're using the pathway of the spinal nerves. So the preganglionic neuron in red is then going to synapse upon the ganglionic neuron in black on the right, and then that will allow for the viscera to be innervated by spinal nerves, because you can see how that ganglionic neuron loops up and around and goes to different sites. So the second pathway here is the collateral ganglia. So the collateral ganglia are things like the celiac ganglion, the superior mesenteric ganglion, the inferior mesenteric ganglion, all of those sites that you can see that are just out lateral to the sympathetic chain ganglia. So these are going to have ganglionic neurons that are going to innervate our abdominopelvic tissues as well as our viscera. And they're typically located just slightly anterior to our vertebral bodies. The collateral ganglia are going to receive sympathetic innervation from sympathetic preganglionic fibers that then are going to synapse in the collateral ganglia that are separate. So let's look at this pathway that's illustrated in figure 16.3. So you can see here how we have our preganglionic fibers in red, and what you're going to note here is they don't synapse in those sympathetic chain ganglia. They just simply pass straight through without synapsing. So those axons are long and they stretch out and then they synapse on the collateral ganglion. So the collateral ganglion include all of the different types of splanchnic nerves. So we have the greater, the lesser. There's 4 different types. But either way, this constitutes the splanchnic nerves. And then from that point on, we have those collateral ganglia, which then have the postganglionic fibers that are able to innervate our visceral organs in our abdominal pelvic cavity. The last pathway here in the sympathetic division is the adrenal medulla, which is totally different. So the adrenal medulla is just the center of each adrenal gland. So the adrenal gland outer aspect is called the cortex, and the inner aspect is the medulla. You're going to find that terminology used in a lot of different organs of the body as we move on, and you'll especially see it a lot in 206 next semester. So this is a modified sympathetic ganglion, and these ganglionic neurons have very short axons. But when stimulated, what happens is they release neurotransmitters directly into the bloodstream as opposed to at a synapse. So because it's released into the bloodstream, that means those neurotransmitters will be dispersed far and wide throughout the body, and they have longer lingering effects because it takes longer for those neurotransmitters to be absorbed and metabolized throughout the entire body. So this gives a longer response. And the adrenal medulla is what's responsible for releasing either epinephrine or norepinephrine. And these will basically function as hormones because they're distributed throughout the body through the bloodstream. So they'll affect all different target cells throughout the body differentially based upon what receptors are present at the different target cells. So some cells may respond to this very differently than others. So here what we can see is we have these long preganglionic red fibers that are extending all the way from the spinal cord. They pass through the sympathetic chain, they pass through or by ganglia, and then you can see that they only finally synapse on the adrenal medulla, which is the center of the adrenal gland right above the kidney. And that is right on the very center of the kidney. So that way, these cells in there that are synapsed upon by the postganglionic neurons are able to release the product out into the bloodstream nearly immediately. So the neurotransmitters that are being transmitted throughout the nervous system, this is how the neurotransmitters become part of the endocrine system. So the take-home of the sympathetic division is that the preganglionic fibers are typically relatively short, and the ganglia are usually located near the spinal cord. So the sympathetic chains, those are right next to the spinal cord. We have the ganglia that are a little bit further out, but they're still closer to the spinal cord. But the postganglionic fibers are going to be rather long, with the exception of at the adrenal medulla, where we have those tiny, short little postganglionic fibers. Because remember, in this case, we have those long preganglionic fibers stretching all the way into the adrenal medulla, and then once they synapse, there's not too far to go in the adrenal medulla. There's only a very small distance. So that's the one exception to this rule. Motor commands that are issued to target structures like the body wall, the thoracic cavity, head, neck, or limbs are going to be preganglionic fibers that are going to synapse in one or more of the sympathetic chain ganglia. So each of the sympathetic chains, remember there's one on each side of the vertebra, are going to contain 3 cervical, 10 to 12 thoracic, 4 to 5 lumbar, 4 to 5 sacral, and 1 coccygeal ganglion in total. Sometimes these numbers are a little bit different because sometimes ganglia that are next to each other can kind of fuse together a little bit, so the numbers aren't exact. Preganglionic neurons are going to be limited to only the spinal cord segments T1 through L2, so that was why we call it the thoracolumbar division. So those spinal nerves are going to have both white rami and gray rami. So the white rami are myelinated, and the gray rami are the unmyelinated postganglionic fibers. The preganglionic fibers are going to be able to innervate our cervical, inferior lumbar, and sacral sympathetic chain ganglia, and those chain ganglia provide all of the postganglionic fibers, so they go through the gray rami, which again are unmyelinated, and then that will go to the cervical, lumbar, and sacral spinal nerves. Postganglionic fibers that are unmyelinated are going to have pathways that are varied depending upon what the target is of this postganglionic fiber. So those that are going to control things like our visceral effectors and our body wall, head, neck, or limbs are going to enter the gray ramus, but then they'll return back to the spinal nerve for redistribution. So this allows for the innervation of sweat glands, the erector pili muscles, and other things as well. But those unmyelinated postganglionic fibers that are there to innervate visceral organs of the thoracic cavity, like the heart or the lungs, those are going to form bundles that are known as the sympathetic nerves. Specifically, in the head and neck, the sympathetic postganglionic fibers that are there will leave the superior cervical sympathetic ganglia, again right next to that vertebral column, and those will supply the regions and structures that are all innervated by cranial nerves 3, 7, 9, and 10. So in summary, the sympathetic chain ganglia are going to have preganglionic fibers from T1 through L2, and this includes the cervical, inferior lumbar, and sacral chain ganglia. Only the thoracic and the superior lumbar ganglia, which is between T1 and L2, will receive the preganglionic fibers coming from the white rami. And every spinal nerve is going to receive a gray ramus that's going to arise from a ganglion of the sympathetic chain. The collateral ganglia, which are next to but a little anterior to the vertebral column, are going to originate as paired ganglia on the left and the right, and they're typically unpaired in adults because these will fuse together oftentimes. So the preganglionic fibers for the collateral ganglia are going to pass through that sympathetic chain without synapsing at all, and in doing so they're going to form the splanchnic nerves that I mentioned earlier. We'll discuss the splanchnic nerves in a little more detail coming up, but these are located in the posterior wall of the abdominal cavity. So the postganglionic fibers of the collateral ganglia leave the collateral ganglia and then they extend throughout the abdominopelvic cavity to innervate our visceral tissues and organs. The whole point of this is that it functions to reduce the blood flow and energy use by the organs that don't need it when there's a short-term survival issue at hand. So again, this is the idea of that you don't need to have digestion going on if you need to be running away from a bear. So this also helps to release stored energy so that way we have the energy to run away if we need to. The collateral ganglia have preganglionic fibers that arise from the 7 inferior thoracic spinal segments, and they end at the celiac ganglion or the superior mesenteric ganglion. The preganglionic fibers that arise from the lumbar segments are going to form the splanchnic nerves, and we have 4 different main splanchnic nerves. These end at the inferior mesenteric ganglion, and all 3 of the ganglia here are going to be named after the arteries that are nearby. So for example, the celiac ganglion, which is a collateral ganglia, it is right next to the celiac artery, which is important for providing blood supply to a lot of our digestive systems, such as the pancreas, spleen, etc., etc. So the celiac ganglion is a pair of interconnected masses that are gray matter, and they're located right at the base of the celiac trunk. And so these can form either a single mass or they can have many interwoven together. But either way, these postganglionic fibers from the celiac ganglion are going to stretch out to innervate our stomach, liver, gallbladder, pancreas, and spleen. Essentially what this means is that the celiac ganglion is the first major collateral ganglia that does the most proximal or the most superior portions of our digestive system. Another example is we have the superior mesenteric ganglion, which is another collateral ganglia. So the superior mesenteric ganglion is going to be located right at the base of the superior mesenteric artery, and therefore it's responsible for innervating the small intestine and the proximal 2/3 of the large intestine. Lastly, our inferior mesenteric ganglion, which is our last collateral ganglia, is located near the base of the inferior mesenteric artery, and so it's going to be responsible for providing sympathetic innervation to our kidneys, urinary bladder, the end segments of our large intestine, and our sex organs. So you can basically break it down. The celiac ganglia is going to innervate mostly the most proximal third of our GI system in general. The superior mesenteric ganglion innervates about the second third, and then the inferior mesenteric ganglion roughly innervates the most inferior third. And this is a very rough estimation, but it's just my easy way of helping you kind of plug it into your mind so that it's easier for you to recall. The adrenal medulla is really neat. So because we're having these sympathetic ganglia at the center of each adrenal gland that are modified, it's a little different than both the collateral ganglia and the sympathetic chain ganglia. In this case, what we have are those long preganglionic fibers that go all the way into the center of the adrenal gland, the medulla, and then they synapse on the cells there. So those cells in the medulla secrete epinephrine, which is also known as adrenaline, and norepinephrine, which is also known as noradrenaline. So typically, the epinephrine is the larger output because it takes up about 75 to 80% of everything that's secreted by the adrenal medulla. But this plays a really important role in our ability to respond to an emergency. So the adrenal medulla is going to have those post-ganglionic fibers that synapse, which release the neurotransmitters that are located there into the bloodstream, so that way those neurotransmitters can be disseminated basically like a hormone. So that will cause changes in the metabolic activities of cells all through the body in different cells contingent upon the receptor on those distant target cells. So this also includes cells that are not innervated by the sympathetic postganglionic fibers. What that means is basically once we release neurotransmitters throughout the bloodstream that go throughout the body, it can affect anything. It doesn't have to be innervated because now it's gaining access through the bloodstream. So the effects of this are really interesting. So instead of just having an action potential fire, and let's say through the motor system, we're able to pick up our cup of coffee because our skeletal muscle had an effector fire and action potential. That makes sense. That's an immediate thing that happens and that's over. But in this case, the adrenal medulla is being stimulated to release its neurotransmitters into the bloodstream, and so then those neurotransmitters are circulating and they're not taken up right away. It may take a little bit of time for this to be absorbed, metabolized, or broken down in any number of ways. So as a result, these hormones will continue to diffuse out of the bloodstream, and the effects last a whole lot longer than a direct sympathetic innervation would. When the sympathetic division of the autonomic nervous system kicks in, this is called sympathetic activation. So the sympathetic division is activated, and this only occurs during a crisis, such as the moment of, I think I'm about to be hit by a car right now. So a crisis like that occurs, and the entire division is going to respond. And this is controlled by the sympathetic centers that are in the hypothalamus, and it affects peripheral tissues and CNS activity both. The changes that occur as a result of sympathetic activation are the exact things we discussed earlier when we discussed what does the sympathetic division of the autonomic nervous system do? Well, it increases our alertness, it gives us that sense of energy and euphoria, so we have infinite ability to run if we need to in an emergency, or the euphoria, which allows us to do something superhuman, like the example of the little old lady who lifted up a car off of her husband when it fell on him when he was changing the oil. You get all of this response through sympathetic activation. Our blood pressure increases, and so does our heart rate, breathing rate, as well as the depth of our respiration. Our muscle tone becomes much more elevated and taut, and we mobilize energy reserves so that way we're able to move very quickly and have the energy to do so. Next, let's discuss section 16-3, sympathetic neurotransmitters and receptors. In section 16.3, we're going to discuss the effects of different types of neurotransmitters and receptors in the autonomic nervous system. So when preganglionic neurons of the sympathetic nervous system are stimulated, acetylcholine is going to be released at the synapses with the ganglionic neurons. The effect of the acetylcholine at that synapse is always going to be excitatory. The ganglionic neurons then that have just been triggered to release their action potential will release neurotransmitters at the target organs at the end of the neuron. And the teledendria that are there are going to branch out and form these networks that create something that looks something like a string of pearls. And each of these quote-unquote pearls is a swollen area called a varicosity, which is the same word that we use for varicose veins at the back of our legs, where we have a bulbous part of a blood vessel. So these varicosities, though, are not blood vessels. These are packed with neurotransmitter vesicles, and so those vesicles then can release their contents, and the membrane receptors are going to be scattered across all different target cells. Figure 16.4 shows you the preganglionic fiber, which is myelinated. You can see the Schwann cells wrapped around it, extending towards the ganglion at the top of the picture. So it synapses on the ganglionic neuron, and then you can see that long purple postganglionic fiber wrapping down to the smooth muscle cells there. So if you look at the zoom-in picture of the varicosities, what we're looking at are all those little purple string of pearls that you see on the muscle cells below are zoomed in upon in that inset image, showing you the vesicles are containing norepinephrine vesicles. Most of the sympathetic ganglionic neurons are going to release norepinephrine, which is abbreviated NE, at those varicosities. So these are called adrenergic neurons. Now there are also some ganglionic neurons that are going to release acetylcholine, so they're called cholinergic neurons. These are located in various places throughout the body wall, in our skin, our brain, and also in our skeletal muscles. The sympathetic nervous system is stimulated primarily from the interactions of norepinephrine and epinephrine with the adrenergic membrane receptors. These can be broken down into alpha receptors and beta receptors, which we're going to discuss a little bit more in the next two slides. Norepinephrine is going to stimulate the alpha receptors considerably more than it does the beta receptors, so it does affect both, but just the alpha receptors considerably more. Epinephrine is going to stimulate both the alpha receptors and the beta receptors pretty much equally, and both of these are G protein coupled receptors. When alpha receptors are stimulated, it activates the associated G proteins on the plasma membrane of the cytoplasmic surface. So we'll break these down to two categories, alpha 1 and alpha 2 receptors. So the alpha 1 receptors are typically found in smooth muscle cells, and so this has an excitatory effect on the target cell. So what this really means is if we have a smooth muscle cell that is excited, it's likely to basically constrict or contract. So if we have smooth muscle in our blood vessels and it constricts or contracts, that will have an effect on vasoconstriction. So this also occurs in blood vessels throughout the body as well as sphincters along the digestive tract, and also this helps our urinary bladder to close. Now the alpha 2 receptors are going to be found primarily on preganglionic sympathetic neurons. So what this means is that we have G protein activation here that's going to lower the cyclic AMP level in the cytoplasm. And so what this has is an inhibitory effect on the cell. So this relaxes things for the most part. So the alpha 2 receptors in the parasympathetic division are going to coordinate both our sympathetic and our parasympathetic activities. But when it's active, the norepinephrine that's included here is going to bind to the alpha 2 receptors, and this is bound at parasympathetic neuromuscular and neuroglandular junctions, inhibiting their activity. So the alpha 2 receptors are overall more responsible for coordination of activities in the autonomic nervous system. Beta receptors are going to be found on the plasma membranes of cells and organs like our skeletal muscles, our lungs, the heart, and the liver. And stimulation of this is going to increase the cyclic AMP levels, and it will trigger changes to our metabolism. We'll break down the beta receptors into three different types. Beta receptors are broken down into three categories: beta 1, beta 2, and beta 3. So when beta 1 receptors are stimulated, we'll have an increase in metabolic activity on those cells. So for example, if we have beta 1 receptors in skeletal muscles, then we have increased metabolic activity in those muscles. It means we're ready to be able to move a whole lot more. And similarly, if we have beta 1 receptors in our heart, it will increase the strength of our contractions as well as the rate of our heart. Beta 2 receptors are going to cause inhibition, so that triggers relaxation as opposed to excitation. So beta 2 receptors can relax smooth muscles, particularly in the respiratory tract. So if they relax, then that allows for the passageways to dilate, so it's easier for more air to come in. Interesting clinical correlation point, by the way. Anybody who has asthma who uses a bronchodilator inhaler is basically triggering this exact pathway, so it allows our respiratory passage to dilate, making breathing more easy. And then the beta 3 receptor is going to be found primarily in adipose tissue. When this beta 3 receptor is stimulated, we'll have lipolysis occur, which breaks down triglycerides that's stored in adipocytes. And when that happens, it's released into our bloodstream for use by other tissues as a source of energy. Most of our sympathetic postganglionic fibers will release norepinephrine, and therefore these are adrenergic. However, a few of them will also release acetylcholine, which makes them cholinergic. So the cholinergic ones are going to stimulate our sweat glands and also expand our blood vessels that go to the skeletal muscles as well as the brain, so we're ready for activity. There are also others, though, that release nitric oxide, which is a gas. So these are nitroxidergic synapses, and these neurons here innervate our smooth muscles and our blood vessel walls. So again, this is an example in our skeletal muscles and also in our brain. And by producing this vasodilation, the effect is that we're able to increase blood flow because there's less resistance with a wider blood vessel. Let's discuss section 16.4, the parasympathetic division, time to rest and digest. The parasympathetic division in many ways is the opposite of the sympathetic division. So the sympathetic division is commonly known sometimes as the thoracolumbar division, and the parasympathetic division will be known as the craniosacral division. So basically it's taking up the two other major components of our central nervous system. So the parasympathetic division has the opposite of the sympathetic division. In the parasympathetic division, we have long preganglionic fibers that are in the brainstem and in the sacral segments of the spinal cord, but then we have short postganglionic fibers in or near target organs. So this is kind of similar to the layout that we had when we discussed the sympathetic division's tract going to the adrenal medulla. In that case, we also had long preganglionic fibers and short postganglionic fibers because the synapsing that occurs in the adrenal medulla doesn't have to go very far. The parasympathetic division is laid out similarly. So these long preganglionic fibers extend out, and the autonomic nuclei that we have that start these are going to be located in the brainstem as well as in the lateral horns of S2 through S4. The ganglionic neurons that are present in the peripheral ganglia are either going to be within or very close by to the target organs, so that way you can affect those target organs easily. As soon as you take a look at the parasympathetic division in figure 16.2, you can see this is totally different than the sympathetic division. So the sympathetic division had a lot of thoracolumbar activity, which is in the middle of this image. As you can see, this is totally absent. In the parasympathetic division, also known as the craniosacral division, we have activity at the top and activity at the bottom of our central nervous system, with the central area kind of being left alone. So as you can see, arising off of the most superior components of our spinal cord and the lower areas of our brain at the pons medulla oblongata, we have nerves coming off that are going to be able to innervate different components of our parasympathetic nervous system. We also have down at the sacral area of our spinal cord pelvic nerves coming off that also innervate a broad variety of pelvic organs. So as you can see here, we have preganglionic fibers in red, and these are going to run very, very long all the way from the spinal cord out towards their final site, and when they synapse on the postganglionic fibers, those are very close to the target organs. So the preganglionic fibers in the parasympathetic division are super long, and the postganglionic fibers here are short, just like the way it was when we discussed the adrenal medulla in the sympathetic division. So again, in the parasympathetic division, we have those really long preganglionic neurons that extend all the way to the site at which they need to synapse. So they're going to synapse in peripheral ganglia that are either going to be terminal ganglion or an intramural ganglion. So a terminal ganglion is going to be near the target organ, and these are usually paired. Alternatively, we also have intramural, and intramural literally means within the wall. So within the actual organ itself, these ganglia are embedded, and this consists of interconnected masses and clusters of ganglion cells. The parasympathetic division is responsible for innervating organs in the cranium, the trunk, and also our pelvic regions. So the way that this is organized is that we have parasympathetic preganglionic fibers, so these are the really long ones. They leave the brain via the cranial nerves. So they come out through cranial nerve 3, which is oculomotor, the facial nerve, which is cranial nerve 7, the glossopharyngeal nerve, which is cranial nerve 9, and our wanderer, the vagus, cranial nerve 10. So these will control our visceral structures in our head, and they synapse in our ciliary, pteropalatine, submandibular, and otic ganglia. The vagus nerve, our wanderer, cranial nerve 10, this is responsible for providing about 75% of all the parasympathetic outflow. So it will innervate structures all along the way as it wanders down in our neck and wandering down to our thorax and our abdominal pelvic cavities, including even a distal portion of our large intestine. The branches of the vagus nerve are going to intermingle with fibers of the sympathetic division because they're going to the same places. In the sacral segment of the spinal cord, the preganglionic fibers, those long ones, are going to be carrying sacral parasympathetic output, and they don't join the anterior roots of the nerve. Instead, they form pelvic nerves, and these innervate the intramural ganglia. Remember, the intramural is inside, within the wall. Intramural ganglia in the kidneys, the urinary bladder, portions of our large intestine, and also the sex organs. Again, in figure 16.2, you can see how the parasympathetic division is the craniosacral division as opposed to the thoracolumbar sympathetic division. And you can see the facial nerve, the glossopharyngeal, the vagus, as well as even up top, the oculomotor nerve, and how those innervate various portions of our body. And then all the way down at the sacral component, you can see arising off of S2, S3, and S4 are pelvic nerves. And the pelvic nerves are all going to extend to everything ranging from our reproductive organs to our urinary bladder and even to our kidneys as well. So what does the parasympathetic division do? It does the opposite of the sympathetic division. So instead of dilating our pupils so that way we can see things really easily, it constricts them. So we just focus on things that are nearby. We're not worried about approaching bear. We're just looking at that plate of turkey and potatoes in front of us. Also, we'll be secreting our digestive gland juices so that way we'll be able to absorb our food better and use all those nutrients that are available to us from what we've ingested. It will also cause changes associated with sexual arousal. So as you can imagine, people can't feel sexually aroused if they think they're about to die, for the most part. So this is part of being relaxed and feeling ready to feel sexually aroused. This also increases our smooth muscle activity in our digestive tract. So again, that's helping with peristalsis of food moving along through our digestive tract. This stimulates and coordinates defecation as well as urination and constricts our respiratory passageways because we don't need to have them dilated to take in as much air as possible when we're relaxing. We just need to breathe at a normal rate. And the same thing is true for our heart rate and the force of the contraction of our heart. That doesn't have to be super strong and super fast because we don't need oxygenation throughout all of our tissues immediately because of the increased metabolic need because we're running from a bear. Because we're relaxing, it's going to reduce our heart rate and also reduce the force of contraction. Time for section 16-5, parasympathetic receptors. So as compared to the sympathetic nervous system, the parasympathetic nervous system can be considered a little bit easier when we discuss the parasympathetic receptors and transmitters. So in the sympathetic system, while we have a number of different types of different substances being released, ranging from norepinephrine, acetylcholine, or nitric oxide, in the parasympathetic nervous system, all we have is acetylcholine, and that's it. So that's pretty straightforward. So the effects of this can vary pretty widely on the postsynaptic cell, and that's due to either the different type of receptor that will accept this acetylcholine, or also on the nature of the second messenger involved that mediates that relationship. So the effect of parasympathetic stimulation on the cholinergic or acetylcholine receptors are localized, and they're only short-lived, which makes sense because this is very different than, for example, when we discussed previously in the sympathetic division, how we had the adrenal medulla releasing its neurotransmitters into the bloodstream, which then become hormones to slowly diffuse into a broad variety of different sites throughout the body. This is going to be localized only. And most of the acetylcholine is going to be inactivated at the synapse by acetylcholinesterase, which is the enzyme that will inactivate acetylcholine. So the acetylcholine that isn't absorbed and inactivated by acetylcholinesterase is going to diffuse into the surrounding tissues, and that gets inactivated by tissue cholinesterase, which is basically the tissue version of acetylcholinesterase that occurs in the surrounding tissues instead of at the synapse. Since we know that the parasympathetic division of the autonomic nervous system only releases acetylcholine as a neurotransmitter, then it's up to the postsynaptic cell to make use of the information that they're receiving. And they do this via receptors. There's also a secondary pathway, and that's based on the nature of the second messenger that's involved here. So first, let's talk about receptors. So cholinergic receptors are all those receptors that receive acetylcholine, and they can be broken down to two categories, either into being nicotinic receptors or muscarinic receptors. So nicotinic receptors are those that are on ganglion cells of the sympathetic as well as the parasympathetic divisions, and they also occur at the neuromuscular junctions of the somatic nervous system. Exposure to acetylcholine is going to cause excitation of the ganglion neuron or the muscle fiber in this case. The other type of cholinergic receptor is the muscarinic receptor, and these are present at either neuromuscular junctions or neuroglandular junctions in the parasympathetic division only. So these muscarinic receptors are G protein coupled receptors, which means that if they're stimulated, the G protein activation will produce longer lasting effects than the stimulation of the nicotinic receptors. So this response can be either excitatory or inhibitory, but it depends on what is activated or inactivated by specific enzymes. A fascinating clinical correlation here is that the names, the nicotinic receptors and the muscarinic receptors, are named this specifically because of research that's been conducted that found that there are very specific environmental toxins that bind to these sites selectively. So the nicotinic receptors, as you might expect, are going to bind to nicotine, whereas the muscarinic receptors are going to bind to muscarine. So nicotine, of course, is found in tobacco, and this targets our autonomic ganglia as well as our skeletal neuromuscular junctions. Nicotine poisoning can occur, and it doesn't happen terribly frequently because it takes a considerable amount, but nonetheless, nicotine poisoning may result in coma or death. So hence, if you do find your child smoking cigarettes when they're young and you think you're going to pull the old school, you're going to sit here and smoke that whole carton sort of thing, that could result in incredible illness, not just an unpleasant experience. So heads up, you heard it here first. And muscarine is a very interesting toxin too. So muscarine is going to affect the parasympathetic neuromuscular or neuroglandular junctions, and both of these are going to produce really dangerous, exaggerated, and uncontrolled responses because our autonomic nervous system is being stimulated so irregularly. So we can have incredible danger as a result of this. Muscarine is really only going to be found in some specific poisonous mushrooms, so it's not something you'll run into terribly often. And also, keep in mind, this targets the parasympathetic division as well. So what you can see is if somebody is poisoned by muscarine, is that they'll have the parasympathetic division completely fired up. So you'll see increased salivation, nausea, vomiting. There'll be constriction of the respiratory passages, so that means not enough oxygen getting in, low blood pressure, and then also a very slow heart rate, which is called bradycardia. So all of these things can occur if exposed to too much of these two substances, specifically nicotine and muscarine. Now, let's discuss section 16-6, the differences in the organization of sympathetic and parasympathetic structures leading to widespread sympathetic effects and specific parasympathetic effects. Before we go any further, let's just look at a really quick summary of the sympathetic division and the parasympathetic divisions of the autonomic nervous system. The sympathetic division is going to have widespread effects throughout our entire body because of the way it's disseminated. So we have two sets of sympathetic chain ganglia, which are those pearls, right, strung along on the lateral aspects of our vertebral column. So that's one of the three pathways. The second pathway is that we have those three collateral ganglia that we discussed previously. And the third pathway is we have two adrenal medulla, one on each kidney in the adrenal gland. So the sympathetic division is going to have those short preganglionic fibers, but post-ganglionic fibers that are quite long. There's extensive divergence, meaning branching in the sympathetic division. And the preganglionic neurons are going to be releasing acetylcholine, and most of the postganglionic fibers are going to release norepinephrine. The effector response in the sympathetic division is going to depend on second messengers involved. The parasympathetic division, in summary, has specific effects on local sites. So the visceral motor nuclei that are associated with cranial nerves 3, 7, 9, and 10 are going to have an effect here, and with S2 through S4. So remember, this is the craniosacral division, and the sympathetic division is going to be our thoracolumbar division. So in the parasympathetic division, we have ganglionic neurons that are located in the ganglia that are either right there within or next to the target organs. And these will innervate regions that are serviced by our cranial nerves and our organs that are in the thoracic and abdominopelvic cavities. This includes 1/5 of the divergence of the sympathetic division, and all of the neurons here are cholinergic, 100%. So the effects are typically brief and restricted only to the specific region affected by the parasympathetic division. Figure 16.5 shows you a comparison between the sympathetic and parasympathetic divisions of the autonomic nervous system. So in both cases, they both start in the central nervous system, although in different locations. So they start with the preganglionic neuron. So those will extend with preganglionic fibers down into the peripheral nervous system. And as you can see in the sympathetic division on the left side, that the preganglionic neuron will synapse at the sympathetic ganglion, and that'll be one of two different places, remember. It will be either in the sympathetic chain or at the collateral ganglia. And so then, or I guess our third option would also be the adrenal medulla, which is illustrated by the ganglionic neuron at the furthest most left, which is then synapsing and putting its product directly into the bloodstream. So if that postganglionic neuron fires and then allows for substances from the vesicles to be disseminated into the bloodstream, that's how we get epinephrine coursing through our veins when we are stressed out and thinking that we might be eaten by a bear. Also, the sympathetic nervous system has that other pathway where once the preganglionic fiber synapses at the ganglion, at the postganglionic neuron, then that will carry information to the target tissue, whichever that may be. Also note, by the way, at the very bottom of the sympathetic image, you'll see that at the bottom of the sample blood vessel that we see carrying oxygenated arterial blood, you'll see that what we have here is green and red arrows hitting up the target tissue, and that's because there are hormones that are in the form of neurotransmitters released from our adrenal medulla, released directly into our bloodstream, so that way they can also affect a distal target tissue, just similarly to the way that a postganglionic fiber could also affect that same tissue. Now, the parasympathetic division of the autonomic nervous system on the right starts again at the central nervous system, and the preganglionic fiber extends into the peripheral nervous system, and then you'll see that it's going to synapse at a parasympathetic ganglion, and then you can see that the postganglionic fiber here is very short. So this is similar to the way it appears when we're discussing the ganglionic neurons in the adrenal medulla. Next, we'll discuss section 16-7, dual innervation. Dual innervation essentially just means that many organs are innervated by both divisions of the autonomic nervous system. So that includes both the parasympathetic and the sympathetic divisions of the autonomic nervous system. And most of our vital organs are in fact dual innervated. So these two divisions, as we've previously discussed, have opposing effects. And the parasympathetic postganglionic fibers are going to travel through the cranial nerves to get to their peripheral destination. Now, the sympathetic innervation will reach the same structures, but they do it in a different fashion. They do it by going from the superior cervical ganglia of the sympathetic chain, for example, to reach the head, whereas in the parasympathetic division, the postganglionic fibers are going to travel through the ciliary, the pterygopalatine, the submandibular, and the otic ganglia by the cranial nerves to get to their destination that is in the periphery. In the thoracic and abdominopelvic cavities, we have autonomic plexuses, and these autonomic plexuses are really just where we have sympathetic postganglionic fibers meeting with parasympathetic preganglionic fibers. So this creates a series of networks of all of these fibers together that merge. And so these are going to travel along with blood vessels and lymphatic vessels to supply visceral organs. So we would call this vessel bundle a neurovascular bundle. Figure 16.6 shows you what the anatomy of dual innervation looks like. So scattered throughout our thorax, abdomen, and pelvis, we have a number of autonomic plexuses and ganglia, including the cardiac plexus, the pulmonary plexus, the esophageal plexus, the celiac plexus, the inferior mesenteric plexus, and the hypogastric plexus. The cardiac plexus and the pulmonary plexus are going to affect the heart and lungs respectively. And so in the cardiac plexus and the pulmonary plexus, we have intersecting fibers that are autonomic, and of course these are both located in the thoracic cavity. So these contain sympathetic and parasympathetic fibers, and they have ganglia that specifically are there to affect these organs. The esophageal plexus is going to contain descending branches of the vagus nerve, our wanderer, cranial nerve number 10, and splanchnic nerves that are leaving the sympathetic chain. So this will include parasympathetic preganglionic fibers of the vagus nerve, and they will enter the abdominopelvic cavity with the esophagus. So these fibers also will enter the celiac plexus, which we'll discuss next. The celiac plexus, which is also known as the solar plexus, is associated with smaller plexuses, such as the inferior mesenteric plexus. And this is responsible for innervating viscera that is inside of our abdominal cavity. The hypogastric plexus, which literally translates to below the stomach, is going to innervate our digestive, urinary, and reproductive organs of the pelvic cavity. So you can imagine the hypogastric plexus as being involved in everything that is in the lower 3/3 of our abdomen and pelvis. So this will contain our parasympathetic outflow to our pelvic nerves, as well as our sympathetic postganglionic fibers that are coming from our inferior mesenteric ganglion. The splanchnic nerves will also come from the sacral sympathetic chain. Autonomic tone is the idea that the autonomic motor neurons have a resting level of activity even when they're not stimulated. And that this is an important function here because if you were to imagine as your skeletal muscle would have a certain amount of resting tone, that then if it's stimulated, it could potentially even amp up even more and increase or decrease activity from there. But with a low amount of tone in the skeletal muscle, you really can't do as much. So the idea with autonomic tone is that you have your nerves maintaining this background level of activity and that can be amped up if needed. So this is a significant point when we have dual innervation, which of course is present in a lot of different sites throughout our body. It's even more important where it doesn't occur. Dual innervation looks something like this. Let's discuss the heart as an example. So the heart is going to be dual innervated, meaning that it will be innervated by both the parasympathetic and the sympathetic divisions of the autonomic nervous system. So when the heart receives dual innervation, it's going to be receiving acetylcholine that's released by the parasympathetic postganglionic fibers, and the parasympathetic division is going to slow the heart rate to wind things down to relax. And norepinephrine that's released by the varicosities of our sympathetic division is going to have the opposite effect. So it will accelerate our heart rate and speed things up. So we have these two things happening at once. And in this case, both amounts of these two different substances, norepinephrine and acetylcholine, are both constantly being released. And so these continuous releases of both norepinephrine and acetylcholine by the parasympathetic and sympathetic divisions of the autonomic nervous system will produce autonomic tone. So when we're at rest, our parasympathetic division is dominating, of course, letting us rest and digest. And if we have some sort of emergency or crisis, well then our sympathetic nervous system is going to kick in and stimulate all of our sympathetic division, and therefore that will inhibit our parasympathetic nerves as well. Some organs, however, are only innervated by one division. So the example that your textbook offers is one of blood vessel diameter being controlled by the sympathetic division. So normally when we're at rest, sympathetic tone is what's going to keep the muscles that are able to either dilate or constrict our blood vessel walls, is going to be keeping those muscles partially contracted, right at around, let's say, 50% of how much they could be dilated to. So if that's going on at a normal point in time, what's happening then is that norepinephrine is being released from sympathetic fibers to the smooth muscle cells in the blood vessel walls, and that's what keeps it at 50%. So that's a normal day we're walking around, no problem. But if more blood flow is needed, then it's only that sympathetic division that kicks in here. There's no effect of the parasympathetic division. So what happens is the sympathetic division can dial things back. So instead of charging things up, the sympathetic system is going to decrease the amount of norepinephrine by decreasing the rate of release. And so as a result of that, the sympathetic cholinergic fibers are going to be stimulated, and the smooth muscle cells that are around the blood vessel walls will be able to relax. So once those smooth muscle cells relax, then the blood vessel will relax too, meaning it will dilate. And so when we have that vasodilation, what happens is we have more blood flow. So this is an example of the blood vessel diameter in our body being controlled exclusively by the sympathetic division, only either increasing or decreasing the amount of norepinephrine release. And therefore, the sympathetic division is completely in control here. Now let's start to pull it together. We are moving into section 16.8, autonomic regulation and integration. As we start to pull things together, let's just reflect for a little bit about the way other parts of our nervous system work. So our somatic motor control is going to be controlled by a number of different centers. All of these are found in all portions of our central nervous system, and this includes things like our lower motor neurons that are going to be found for our cranial and spinal reflex arcs. And also this includes our pyramidal motor neurons that are in the primary motor cortex. So these somatic motor controls are kept in place by simple reflexes that are all based in the spinal cord, and this allows us to be able to respond really quickly to whatever stimulus it is. And so this is without rising to the level of our consciousness. We have all of these great reflexes. There are centers in the brainstem, however, that are going to control the activity of the sympathetic and parasympathetic divisions, and that will help to modify and regulate these other actions that are occurring throughout our body as well. Just as we have reflexes that affect our musculoskeletal system, we also have reflexes that affect our visceral organs. So these are called visceral reflexes. The visceral reflexes are controlled by the autonomic nervous system, which makes sense because they're not going to be controlled by us willingly or voluntarily. And they have polysynaptic reflexes, meaning that there are multiple synapses that occur in a reflex arc in the viscera. Also, they're of course all initiated in the viscera itself. So this allows for automatic motor responses, and these automatic motor responses can be modified, facilitated, or inhibited by higher centers such as, and especially, the hypothalamus. So a reflex arc in the viscera here is a little bit different, but similar in some ways. So what we have, of course, is a receptor that's able to sense some need to change to a certain stimulus. That sends an action potential along a sensory neuron. So the sensory neuron then will synapse at a processing center consisting of one or more interneurons. And then that will synapse again on two visceral motor neurons. So we're going to look at this in a moment after we talk a little bit about two different types of visceral reflexes. Visceral reflexes can be broken down into either long reflexes or short reflexes. So long visceral reflexes are basically the autonomic equivalent of a polysynaptic reflex. So in this case, we are involving the CNS in the long reflex, and by comparison, the short reflex bypasses the central nervous system completely. So in a long reflex, we have visceral sensory neurons that are going to take information that travels along the posterior roots of the spinal nerves, and it comes to the central nervous system. So these visceral sensory neurons are going to be housed within sensory branches of cranial nerves, and also within autonomic nerves that innervate visceral effectors. So the autonomic nervous system will be able to take this information and carry the motor commands out to the visceral effectors to make a change. So the long reflexes take the central nervous system and this long process by which they're able to coordinate the activities of an entire organ altogether. Now the short reflexes bypass the central nervous system completely. They just have a sensory neuron and an interneuron, and the interneuron cell bodies are going to be housed in autonomic ganglia, which are outside of the central nervous system. So the interneurons there, which are the processing centers, are going to synapse on the postganglionic neurons, which then will have the postganglionic fibers extend to distribute out the motor command. So this allows for a very simple motor response with only localized effects. So a short reflex is going to be able to control activity only in one small part of the target organ, whereas the long reflex coordinates activities of an entire organ and also involves the CNS. Figure 16.7 in your text shows you the difference between what a short reflex looks like and a long reflex looks like in a schematic. So first, let's start off with a short reflex. So starting at the top left corner, you see the arrow that says stimulus. So that's where we'll begin. So we have receptors in these afferent sensory fibers that will sense the stimulus. So those will then synapse at an autonomic ganglion, which is either sympathetic or parasympathetic. And so it'll synapse there at that interneuron, which is shown in white. And then that will synapse on the postganglionic neuron in black, which will send out a response immediately to a peripheral effector. So the short reflex is able to bypass the central nervous system. It's fast, and it innervates only a very small area within a certain organ. Now, the long reflex is a little different. So we'll start off again at the top with the stimulus at the left, and we have our receptors in the peripheral tissue, again, which are on our afferent sensory fibers. And so that will synapse, instead of synapsing outside the central nervous system at a ganglion, it's going to synapse actually in the spinal cord in a processing center. So it will synapse onto an interneuron in the spinal cord, which then sends the information on to a preganglionic neuron, which is shown in red. And then it will synapse at the autonomic ganglion, which again could be sympathetic or parasympathetic, contingent upon the control required. And then it will synapse again finally on a postganglionic neuron, shown in black, which sends the information out to the peripheral effector. So the long reflex is involving the central nervous system because it goes to the spinal cord and is able to control an entire organ. In general, our long reflexes of the autonomic nervous system are going to be important in regulating the visceral activities in most of our organs at an organ level. Our short reflexes, though, are going to provide more control and coordination in things like the digestive tract, in which case, if you think about the digestive tract, we have a giant long tube, and we don't want it all contracting at the exact same time. That would be silly. Nothing would happen, right? We need a sequential series of contractions in order to propel food along. So we need these short reflexes that can just innervate specific segments, and also there are associated glands in these segments too. So neurons that affect the enteric nervous system are going to be composed of short reflexes. Let's discuss this next. As a quick review, the way that we break down the nervous system into functional divisions is first into either afferent divisions or efferent divisions. So are we having sensory information being brought into the central nervous system, or are we having information being sent out peripherally via the efferent division? Another way to break this down is we take the efferent division and we look into their two divisions, the somatic nervous system, which is our skeletal muscle, and this is all voluntary, so we discussed this in a previous chapter. And then we have the autonomic nervous system, or the visceral motor system, which we're discussing in this chapter today. So there's a third division, and that is the enteric nervous system. And this is just a whole network of neurons and nerve networks that are in the digestive tract that allow for a coordination of multiple local complex visceral reflexes. So the enteric nervous system has ganglia that are present in the walls of the digestive tract that contain the cell bodies of a whole number of these different types of neurons. We have visceral sensory neurons, interneurons, and the visceral motor neurons having all of their cell bodies in that ganglia. So the axons then branch out extensively to form these nerve nets that are able to control the digestive functions totally independent of the CNS. So in this way, we have these axons stretching out, and so we're able to then innervate a segment at a time so that we can contract sequentially to have peristalsis. When we have more complicated sympathetic and parasympathetic reflexes, will require a little additional support. So this is where we have processing centers and nuclei in the medulla oblongata that can help to coordinate these complex reflexes. So in the medulla oblongata, we'll have centers and nuclei that are involved with things like salivation, swallowing, our digestive secretion, peristalsis, and urinary function. And then further, all of those centers and nuclei are also able to be regulated by the hypothalamus. As you can see in figure 16.8, we're comparing somatic and autonomic function. And in this image, we've divided the central nervous system in purple above. And you can see the cerebral cortex is the very top of this image because this is the highest level of pulling things together. And then at the bottom in yellow, we have the peripheral nervous system. So all of our sensors and effectors are out in the peripheral nervous system, and you can see that there's a number of different pathways that are interconnected here. And the more complex we go, the higher up we go in the central nervous system channels or layers that you can see here. So this image is showing us the integration of the autonomic nervous system and the somatic nervous system activities. And you can see there's a lot of parallels in the way things are organized and the way things function. But the key take-home here is that integration of these functions is going to occur at the level of the brainstem, and that both the autonomic nervous system and the somatic nervous system are under control of higher centers, as you see here. So just very briefly, if you look at this image at the bottom center, you see sensory receptors. So when we sense something, regardless of where it is in our body, we'll have to have some information go somewhere to make some sort of response to it. So if it's going to be something that involves our autonomic nervous system, on the right we have short reflexes and long reflexes. And you can see how the short reflexes stay in the peripheral nervous system with a ganglionic neuron there that it will then send information to a visceral effector. Alternatively, if this is a more long reflex that goes into the central nervous system, you can see the next arc is up in the purple, and then that will go back down through the autonomic nervous system to the visceral effectors. Now alternatively, let's look on the left side of the image. if we sense something of our skeletal muscle system, we have the same sort of sensory pathway, so it goes up through the center, and then we'll switch over to the left for somatic reflex. So in this case, we'll bring information back down through the somatic nervous system out to our skeletal muscles to be able to move away from something hot, for example. Now, those are all the simple, straightforward types of reflexes. If we have something more complicated or complex that we need to process, in that case, the information has to be raised up to the level of the brainstem, which you can see is a big, broad swath across the whole entire plane at the central nervous system that nothing gets past it, basically. So the brainstem is the integration center of both the autonomic nervous system and the somatic nervous system. After that, we have a number of different components of our central nervous system that will be involved, such as our thalamus, our limbic system, our hypothalamus, and most notably our cerebral cortex. But please take away from this image that our integration is occurring at the level of the brainstem, and this is the largest component in this schematic, trying to illustrate that very point. Now let's discuss section 69, higher order functions. Our higher order functions are things like our memory and the states of our consciousness. And so these are going to share three characteristics in common. So the first is that every higher order function will absolutely require involvement of the cerebral cortex. So in order for anything to be performed that would be considered a higher order function, we have to have a complex interaction between different areas of the cortex and other areas of the brain. Now this also includes conscious and unconscious information processing. And further, these are subject to adjustment over time. So these are not things that we are born with, such as innate behaviors or things like that. This is something that is learned, that can be adjusted, and over time can become slightly different. So we all understand what memories are, but a definition of a memory is kind of helpful here. So a memory is really just stored information that we've gathered through experience. It's not something that we instinctively do or reflexively react to. This is something that we have put into our brains through experience in the world. So there are two different types of memories. There are fact memories and there are skill memories. So fact memories are just bits of information, like that you know, for example, that there are 12 months in a year, for example. Now, skill memories are going to be much more different and difficult to be able to acquire. These are things you actually learn and have to work hard to learn sometimes. So these include things like motor behaviors. How do you pick up a pen and write, for example? How do you use a spoon to bring food to your mouth? Which is apparently very complicated because my toddler seems to have to work at this quite a bit. And these are things that become incorporated at an unconscious level after doing it repeatedly over and over again. So the brainstem is going to store these related to innate behaviors, and these complex skill memories are going to integrate different motor patterns in our basal nuclei, in our cerebral cortex, as well as in our cerebellum. Now we'll discuss short-term memories versus long-term memories. And I am sure you've all thought of this before because this is the sort of thing you think about when you're studying, hoping to remember things before an exam. So short-term memories are things that you can hang on to and recall, and you can recall them immediately right after, but they don't last long. So there are small bits of information here, and you can repeat this information over and over, and by doing that, you can allow your short-term memory to be converted into long-term memory, and this is through a process called memory consolidation. So this is one of those reasons why in every class you've probably ever taken, you've had your professor say, read the text 3 times, go through your notes several times, read it before you go to bed at night, read it again when you wake up in the morning. That way you're taking those short-term memories and converting them to long-term memory. And this is particularly important when you go to take an exam because you can read the textbook on Monday and think, yep, I get it, I remember, and then you go to take your exam the following Monday, and those short-term memories are gone. So you have a false sense of security thinking that you know it because the week before you absolutely recalled, but you didn't consolidate that information from a short-term memory into a long-term memory. So next, let's talk about long-term memories. Now, long-term memories get broken down into 2 types. There are secondary long-term memories and tertiary long-term memories. So the secondary long-term memories are going to fade with time, and they may require some effort to recall. So this is the sort of thing that you have to think back, geez, where was I? Where did we have that birthday party when I turned 16? Something like that. Now, a tertiary memory is something that is so emblazoned into your mind that you will never forget it. It's something like your mother's face, or your favorite song. Things like that that get really attached to you are your tertiary memories. So what we're hoping when you take any course like anatomy and physiology or anything like that is that ideally you will develop long-term memories, and we don't expect necessarily that they will become tertiary memories that you never forget for the rest of your life, because you certainly will forget a lot of the things that you're studying here. And in fact, if you speak to anybody who's a healthcare professional, they won't remember some of the things that you are studying right now. Yet they can still do their job. And why is that? Because they have secondary memories of what they've learned in school. So while information may fade with time and it might require them some effort to think about it, when the situation arises, they know what they need to look up, they know what they need to look into, so that way they can ask the right question to do the right thing. It's not that they have every single bit of information that they've ever learned in school right at the fore of their mind. So those secondary memories are really important for you to use to be able to be just a really great member of the healthcare team and to be able to know what kind of questions to ask. You're not expected to always know everything, but you do need to know what to look into and what questions to ask. Figure 69 shows you the process by which a short-term memory can become a long-term memory. So if you have some sort of sensory input, for example, you're trying to recall the difference between the autonomic nervous system and the somatic nervous system, you can repeat it over and over and over to yourself, and that will promote retention, so eventually it will consolidate into a long-term memory. Now that said, that short-term memory could fail through a number of reasons. So it could fail through neural fatigue or shock or interference by any other stimuli. So for example, you're trying to learn this chapter, but something emotional happens in your life that's considerable. For example, somebody gets very ill. You're going to be thinking about that more than you're going to be thinking about what you're studying, right? So this is a way in which short-term memory can be lost if there's interference by other stimuli. On the other hand, if you do consolidate the memory, if you manage that, then you have long-term memories that form. And remember, we have secondary memories and tertiary memories, which are long-term memories. So the secondary ones are going to be those in which they don't last long and you can recall them right away. So this is getting up first thing in the morning and reviewing your notes before you take your exam. You'll have some long-term memories that will be more freshly accessible. Now the tertiary memories, on the other hand, those ones you will have there forever, right? So they are going to be built into the background of your mind. You will never ever forget. So hopefully, if you consolidate well and continue to review material, you'll have those tertiary memories with you for as long as possible. So the cerebral cortex is going to be the home of fact memories, and then the cerebellum is also involved when we talk about skill memories, because the cerebellum helps us to coordinate our motion and movement. Taking a memory from a short-term memory and consolidating it into a long-term memory is going to require several different parts of our brain. The amygdaloid body and hippocampus, which are both parts of our limbic system, are involved, as well as the nucleus basalis and the cerebral cortex. We'll discuss those more in the subsequent slides. The amygdaloid body and the hippocampus are both components of the limbic system, and these are absolutely essential to memory consolidation, which is the process of taking a short-term memory and having it become a long-term memory. So if there's damage to the hippocampus, then that will cause the inability for you to be able to take any short-term memory and convert it to a new long-term memory. Now, that doesn't mean that any old memories would be affected. If you have an existing long-term memory, then those would remain intact and totally accessible. It's just that you wouldn't be able to learn anything new and retain it indefinitely. The nucleus basalis is a cerebral nucleus that's located near the diencephalon, and the role that it plays in memory storage and retrieval is uncertain. But we know that tracts of the nucleus basalis are going to connect with the hippocampus, the amygdaloid body, and all parts of the cerebral cortex. So damage to this nucleus is going to change our emotional state, our memories, and also it can interfere with our intellectual functions. The cerebral cortex is the location of storage for most of our long-term memories. So our conscious motor and our sensory memories are going to be referred to the appropriate association areas. So for example, that would mean that if we have visual memories, that those would be stored in the visual association area. Now, portions of our occipital and temporal lobes are also involved, and these are critical to memories of our faces that we see, voices we hear, and the words that we hear and use as well. Further, a specific neuron in itself may be activated by only hearing one word or even by a specific combination of sensory stimuli. The cerebral cortex, being home to most of our long-term memories, will have all sorts of different association areas that will allow us to store different pieces of information in different places. So for example, our visual association area will allow us to be able to recognize what something might look like, or that the letters of a specific word spell out what something means. Our auditory association area will take in things like the sound that the book gives the example of a cow, right? So if a cow moos, when you hear the sound moo, it's the auditory association area that recognizes that that's the sound that a cow makes, for example. It also recognizes the way the word cow sounds. So when somebody says the word cow, your auditory association area recognizes what you hear and can interpret that when someone says cow, they're referring to that big black and white Holstein that you've seen before. The speech center will also tell you how to say the word cow, for example. And then in our frontal lobes, we'll have even more detailed things about what we know about cows to be true, for example. That they tend to hang out in friends, that they have long memories, that they're able to hold a grudge and be excited when they solve a problem. All these things are true, and they're stored in different parts of our cerebral cortex. So if a specific storage area gets damaged, then the memory associated with that area will be incomplete and potentially ruined. The consolidation of memory at the cellular level is going to have both anatomic and physiologic changes at both our neurons and synapses. We'll discuss these in the coming slides, but the three things that happen in general are we have increased neurotransmitter release, there's facilitation that occurs at the synapses, and we form additional synaptic connections. A synapse that's frequently active will increase the amount of neurotransmitter that it stores. So therefore, on each stimulation, more and more of that neurotransmitter is going to be released. And then the more neurotransmitter is released, then the greater effect that that's going to have on the postsynaptic neuron. So if we have a neural circuit that has repeated activation over and over, that will result in that continuous ongoing release of neurotransmitters being released over and over. And that neurotransmitter then will bind to receptors on the postsynaptic membrane. So the more that this occurs, it will create a graded depolarization that continues to bring the membrane closer to threshold. So this is called facilitation, as we've discussed before, and this resulting facilitation is going to affect all of the neurons that are in this specific circuit. The third cellular mechanism of memory formation and storage is developing new synaptic connections. So if one neuron continues to repeatedly communicate with another one, then the axon tip of the presynaptic neuron is going to branch out and form additional synapses on the postsynaptic neuron. So what this means is that that presynaptic neuron now has a greater opportunity to affect the membrane potential of the postsynaptic neuron. I think of this like if you're watering a plant. The more that you water a plant, the roots will grow towards the water, right? Because they want to get that connection and bring that in. Similarly, when neurons repeatedly communicate, they're going to grow towards the synapse that they're going to be synapsing upon. Those three steps that we just described are all facilitating the communication along a specific neural circuit. So this will be caused by anatomic changes, specifically including the addition of new growths of new axon terminals to be able to synapse upon the postsynaptic neuron. And this is considered to be the basis of memory storage. So a memory engram is just a single circuit from start to finish that corresponds with a single memory. So the idea is that you have a specific memory located in a specific portion of your brain, and the pathway to get there is a memory engram single circuit. So this will form as a result of experience and particularly as a result of repetition. So this is one of the reasons, again, why your professors always nag you to read and reread, etc., etc. Also, it's worth noting that a memory engram is going to take at least an hour to form. So it means that likely if you read something once and don't think about it again, you're probably not going to keep a permanent recollection of that memory. It's worth reviewing several times. That way you can develop this circuit to have this memory become a consolidated long-term memory. There are many factors that will affect the formation of memory engrams. So for example, the nature, intensity, and frequency of the original stimulus will all play a role in our ability to form a new memory engram. Also, there are drugs that can stimulate our central nervous system. Hello, caffeine in our coffee, or nicotine in cigarettes. These things can actually enhance memory consolidation, as much as I hate to tell you that. Nicotine, of course, as we know, has multiple negative and detrimental health effects. However, caffeine in moderation can be one of those things that's actually very helpful. So have a cup of coffee when you're studying. I'm not recommending having a cigarette, but have a cup of coffee and not too much of it, by the way. Also, our hippocampus is involved in this as well, but there's no known particular mechanism. So some research has shown that it's linked to NMDA receptors, and blocking these receptors prevents long-term memory formation. But more research needs to be done on this particular area of study. When we consider consciousness, it's pretty easy to just think about the two far opposite ends of the spectrum, conscious and unconscious. But there's a lot of shades of gray that vary in between those two. So for example, consciousness can range from anything from being just barely waking up to really, functioning normally to feeling like you're at your peak time of the day where you're ready to learn. So for example, I'm a morning person. Some of you might be night people. So that might be your peak time of maximum consciousness and activity. But also you can make yourself very jumpy and twitchy if you drink too much caffeine. So again, just take it easy with the caffeine. But you can also have a range of unconsciousness as well. So if you're just falling asleep, for example, you might wake up if you hear a commercial on the TV or, if the dog walks by, for example. Or you could be in the middle of a full deep sleep in which it'll take a little bit more to rouse you. And even further to that effect, if somebody's in a coma, then they'll be unconscious and you cannot awaken them even with a strong stimulus. So you could poke people, you can try to say hello, talk to them, shake their arm. In a coma, all of these things are not going to stimulate them adequately to awaken them. So when our central nervous function becomes abnormal or depressed, then our state of wakefulness is going to be affected similarly. During the state of deep sleep, which is also called slow wave or non-REM sleep, which stands for rapid eye movement, our entire body will be able to relax. So this is the point at which we get maximum restoration to our body through sleep. Our cerebral cortex activity comes down to a minimum, and our heart rate, our blood pressure, our respiratory rate, and our energy use all decline considerably, even up to 30% of normal. It's during rapid eye movement sleep, or REM sleep, in which we have our dreams. So this is when we have considerable change in our blood pressure and respiratory rate in response to what we're dreaming about. Our EEG, or electroencephalogram, will resemble that of an awake state during REM sleep. And during this time, we're less receptive to outside stimuli than in deep sleep, but we're still able to respond in some fashion. We can be awakened, for example, more easily. Our muscle tone is going to decrease markedly from our awake state, and we'll also have considerable inhibition of our somatic motor neurons. Not entirely, mind you, because we do sometimes move a little bit while we're dreaming. And during rapid eye movement sleep, the signature component of this is that our eyes, in fact, do move very rapidly. During sleep, we're going to alternate back and forth between REM, or rapid eye movement sleep, and deep sleep. So as we start off sleeping, the REM periods will be about 5 minutes or so on average, but then over the course of the entire sleep, it'll increase all the way up to 20 minutes. While we're sleeping, there are a variety of different changes that occur in our bodies, including some slight physiologic changes in the activity of our organs and organ systems, but none of these are considered to be essential changes that are imperative for the healing or maintenance or function of those organs or organ systems. So we do know that protein synthesis in neurons increases, and so it appears, so far anyway, with the current understanding of science in regards to sleep, that the importance of sleep is generally localized to the central nervous system, and that our central nervous system truly does require sleep to function optimally. And I think that that makes sense to most people. So we know that extended periods without sleep are going to lead to disturbances in our mental function. So if anybody out there who's listening has ever experienced any type of insomnia, I'm sure you know exactly like when Edward Norton says in Fight Club that when you have insomnia, when you try to sleep, you can't sleep. But when you're awake, you're not really awake either. So we absolutely need sleep in order to have our central nervous system repair, heal, and allow us to form new memories as well. Figure 1610 shows you what different EEGs look like for people who are either awake in REM sleep versus deep sleep. So you'll notice right away as soon as you look at this that the top line showing you the awake EEG and the second line showing you the REM sleep EEG, those look remarkably similar. So we know there's a lot going on in our brains when we're in REM sleep. But it's during deep sleep that we actually have a completely different EEG. You can see that that looks nothing like the typical awake alpha waves for awake adults. Figure 1610, the second part of it, shows you what a typical pattern of sleep looks like for a healthy, normal young person during a single sleep. So as you can see here at the top of the graph, we have our awake period, then there's REM sleep, and as we move further down the axis, you see we go into a transition period in between REM sleep and deep sleep. And you can notice that we only really get into deep sleep a couple times at the very beginning of our sleep. So as we first fall asleep, we get a lot of really great rest right then. And then as the sleep goes on, we get less and less deep sleep. And we have longer and longer REM sleep, as you'll notice. Those peaks in the pale blue area indicating REM sleep start off being just a very narrow little peak right around 11 p.m. through midnight. But then, as you see around like 3 in the morning and 4 in the morning, those peaks there are considerably longer periods of time in REM sleep. The reticular formation appears to be the component of our brain that allows us to be aroused and awaken from sleep in the first place. And the reason for this is believed to be because of all of the extensive interconnections in between the reticular formation with both sensory and motor components of our brain, as well as integrative nuclei. There are also pathways along the brainstem that play a role as well. Now, this is determined by complex interactions between the reticular formation and the cerebral cortex and is still being studied. The reticular activating system is a component of our brainstem that plays a really important role in our ability to be awakened after sleeping. So the reticular activating system is going to extend from in the medulla oblongata to the midbrain, and the output of the reticular activating system is going to extend to the thalamic nuclei, and those thalamic nuclei are responsible for influencing large areas of our cerebral cortex. So it's through that relationship that the reticular activating system affects the cerebral cortex. So when the reticular activating system is inactive, then the cerebral cortex has to be as well, right? And stimulation of the reticular activating system is going to produce widespread activation of the cerebral cortex via that thalamic nuclei. If we're stimulated while we're sleeping, if it reaches the level of activating the reticular formation and the reticular activating system, then we'll be aroused and awaken quickly. And the effect of a single stimulation of the reticular activating system can last for less than a minute, but then after that, then our consciousness is not maintained by anything from the reticular activating system, it's maintained by positive feedback from our cerebral cortex, basal nuclei, and other sites as well. To regulate our sleep and wake cycles, we have essentially dueling nuclei located in our brainstem, which kind of function in a similar way to the sympathetic and parasympathetic nervous system, if you want to use that model in your mind as an example. So there are different nuclei that are using different neurotransmitters. One will try to keep you awake, and one will promote deep sleep. So one group is stimulating the reticular activating system with norepinephrine. So of course, epinephrine is a stimulant, right? So that norepinephrine maintains our awake and alert state. But then there's another group that will depress the reticular activating system via serotonin. So that will help us to have a deep sleep and relax. So these dueling nuclei in the brainstem have an important role in regulating our sleep and wake cycles. Figure 1611 shows you the reticular activating system, which is a diffuse network in the reticular formation and extends from the medulla oblongata to the midbrain. So you can see here the reticular activating system is the midbrain headquarters here of the reticular formation. So it's receiving collateral input from a whole variety of different pathways that are sensory. So in this area, the reticular activating system is stimulated, it will produce arousal, and this will heighten our state of attentiveness and alertness. The chemistry occurring in our brains has a huge impact on how our brain functions, which allows us to behave in certain ways. So if there are changes in the levels of the neurotransmitters present in our brain, that can really affect how we function. So one such example is Huntington's disease, in which case the acetylcholine secreting and GABA secreting neurons in the basal nuclei are totally destroyed. So what happens is as those basal nuclei and frontal lobes degenerate, we start to see more symptoms kick in. And these symptoms that occur are things like we have difficulty controlling our movements, so we see irregular movements come up. Also, intellectual abilities will gradually decline as well. Your textbook describes 3 different neurotransmitters and how that affects brain chemistry as well as behavior, the first of which is serotonin. So serotonin is delivered to nuclei and higher centers in our brain through an extensive network of tracts, and this affects our sensory interpretation as well as our emotional states. So you've probably already heard about serotonin and its role in depression. So a type of drug called an SSRI, or selective serotonin reuptake inhibitor, is a drug that is trying to address this serotonin pathway. So what fluoxetine or Prozac does is it will slow the removal of serotonin at the synapses, which will effectively cause an increase in serotonin concentration at the postsynaptic membrane. So by slowing that down, we're actually basically increasing the availability of serotonin. Another way to look at this is that serotonin can have the effect of producing hallucinations if the serotonin receptors are overly stimulated. So LSD, a drug that was very popular in the 60s, has that very effect. It activates serotonin receptors considerably in the brainstem, the hypothalamus, and in the limbic system, and that can create all sorts of different hallucinations. So these SSRIs have actually been a medical breakthrough. If you're ever interested in reading a really good book about this, I read one when I was 16 because I was that kind of girl. The textbook called Listening to Prozac discusses how this was a breakthrough in terms of treating depression and anxiety. Another neurotransmitter that affects brain chemistry and behavior is dopamine. If we have an inadequate level of dopamine or too much dopamine, these can all result in considerable functional problems for us. So for example, if we have inadequate levels of dopamine, then we end up with Parkinson's disease, so we have considerable motor problems. On the other hand, if we have excessive production of dopamine, that can be associated with schizophrenia, and amphetamines, which are basically going to stimulate the secretion of dopamine, have the same effect in which they can create schizophrenic-like behaviors. So dopamine is important in nuclei that control intentional movements. It does have incredible value as a normally occurring neurotransmitter, but it's when we have either an excessive amount or an inadequate amount that we end up with other problems. Last, let's look at section 1610, age-related changes in the nervous system. Now we'll discuss the effects of aging on the nervous system. This section is not going to leave you feeling terribly positive, but it is important for you to understand these processes. So the effects of aging are basically that everything starts to break down eventually. And by the time we're age 30, we're going to have some considerable anatomic and physiologic changes occurring. These will accumulate over time, and they accumulate such to the effect that by the time we're 65 years old, about 85% of people are going to have changes in their mental performance and their central nervous system function. And when I say changes, what we're really meaning is a decrease in performance. So it's really going to occur to pretty much everyone eventually. Some of the anatomic changes we expect to see related to age in the nervous system include a reduction in brain size and weight, as well as a reduction in the number of neurons. So this is interesting. The brain size and weight decreases as a result of the volume of the cerebral cortex decreasing. And so elderly people are going to have more narrow gyri and more wide sulci, and then a larger subarachnoid space. So that's just a complete reduction in volume at the periphery of the brain at the cerebral cortex. There's also a reduction in the number of neurons, and however, this brain shrinkage isn't related necessarily to a loss of neurons in the brainstem nuclei, but it is linked to loss of cortical neurons. Additional age-related changes that we see in the nervous system include a decrease in blood flow to the brain and changes in the synaptic organization of the brain. So over time, what happens is we'll have fat in our bloodstream that will deposit on the intimal lining or the inner wall of the blood vessels. This deposit is going to narrow the lumen, and in doing so, it reduces the blood flow through the arteries in a process called arteriosclerosis, also known as atherosclerosis. This increases our chances of a cerebrovascular accident, or CVA, which is commonly known as a stroke. There are also changes in the synaptic organization of the brain. So we lose synaptic connections, and also then the rate of neurotransmitter production declines, which is in sharp contrast to as we learn something and we create new connections through the repeated expression of neurotransmitters, those axon terminals will branch out and try to create new connections. In this case, we're having the exact opposite thing happen. So we lose those connections, and neurotransmitter production is going to decline subsequent to that. Further, there are intracellular and extracellular changes in the central nervous system as a result of aging. So the neurons in our brain are going to accumulate some intracellular deposits, some of which are called lipofuscin, which is a granular pigment that has no known function but is commonly found in the aging brain. There are also neurofibrillary tangles, and these are just neurofibrils that are kind of matted together into a big mass, and so these will exist inside the cell body and the axons. Plaques are going to be when we have an accumulation of these fibrillar proteins that are outside of the cell, and that's surrounded by a number of abnormal dendrites and axons. Now, applying to both plaques and tangles, these both contain deposits of several different peptides, and they're primarily two forms of amyloid protein, specifically amyloid A. These will appear in brain regions that are associated with memory processing, so you can imagine how this could affect our ability to recall things. So here's the quick laundry list. All of these anatomical changes that we just discussed will lead to functional changes because structure creates function, right? So we have our memory consolidation becoming more difficult, so it's harder to learn new things and to recall it. We have secondary memories becoming harder to access. So these are things you once knew about the street you lived on when you were 5, for example. These sorts of things which are secondary memories that have been consolidated, they're not quite tertiary memories. They're secondary memories and they're harder to get access to. All of our special senses become less acute. Our reaction times are slowed down. Our reflexes will weaken and even sometimes disappear entirely. Our motor control becomes less precise. And further, the motor patterns that we used to do very quickly and easily will take a little longer to actually complete. It's worth noting that although we have a whole variety of age-related changes occurring in our central nervous system, that even though this is occurring, it doesn't interfere with people to live and have a totally normal life, right? We all have different little things that we deal with, ranging from migraine headaches to a bad tooth to, a torn ACL. We all have different things that go wrong with us and our bodies betray us all in time. But we can function anyway because we're able to maneuver around these different issues. Some individuals, however, will develop senile dementia, which is also called senility, and this will end up including some things like memory loss, anterograde amnesia, which is the ability to make new memories, and also having emotional disturbances. And of all the different types of senile dementia, the most common is Alzheimer's disease. Thank you so much for your attention, everyone. I realize at this point in the semester, you're probably wearing down a little bit. You'd probably like a break, but you have to keep going. You have to keep burning that candle at both ends and keep grinding through. You are almost at the end. You are almost there. You have almost finished Bio 205 online in a pandemic. I am so proud of you. You're almost there. So keep your eyes on the prize, keep focused. And in the meantime, I thought you might like to see something adorable. This little guy that you see here is Fitzroy. He is the newest addition to my family, and he's an Old English Sheepdog puppy, and he's very bad. He does all sorts of naughty things, but he's a very sweet guy, and we're in love with him. So I just thought you might like to see a cute little friend, and I hope that that might cheer you up. Anyway, back to studying, back to A&P. Have a wonderful week.